Alright, for our discussion in section 15.2, we're going to be looking at some linkage going on between selected genes. First off, we're going to be looking at sex-linked genes. So, looking right here, the chromosomal basis of sex, we can see that this image shows us that males have 44 chromosomes or autosomes in our somatic cells along with an X or a Y chromosome, and that females have 44 autosomes along with an X and an X chromosome, and that we can form gametes of one of three varieties depending on gender here. Males, we can form some gametes that are 22 autosomes and an X chromosome, or we can form gametes that are 22 autosomes and a Y chromosome. Females is a little bit different. You're either going to get 22 chromosomes and an X, or you're going to get 22 chromosomes and an X. Looking at this image here on the screen, I have no idea why this is 22 plus XY. That's not possible coming from a female. It should just say 22 plus X. That Y should not be there. Anyhow, when we recombine these, sperm with the ova, depending on which sperm happens to fertilize to form the zygote, we'll either have a new zygote with 44 autosomes and 2 copies of the X chromosome, or a zygote with 44 autosomes and a copy of an X and a copy of a Y chromosome. This is known as the XY system, which is what we see going on with mammals, specifically humans. But that's not always the case with all living things. We can see that these crickets here work on the XO system, where this O is actually a 0. Females will be 22 autosomes and an X and an X chromosome, and males will be 22 pairs of autosomes and a single X chromosome. That's it. Chickens, roosters work on the ZW system. Females will have 76 chromosomes along with a Z chromosome and a W chromosome, which are their sex chromosomes, whereas males have the 76 autosomes and 2 copies of the Z sex chromosome. And then we have the haplodiploid system, where homologous chromosomes don't necessarily exist. We can see in females here that they have 16 chromosomes, and those chromosomes are homologous, putting that cell, or female cells, in a diploid state. But males, on the other hand, are all haploid, so they have 16 total chromosomes. There is no homologs present. So that is the haplodiploid system. So moving along, the chromosomal basis of sex, we know that the X and the Y chromosomes, they're not homologous in humans, but they are similar in that they determine sex traits and genes. And we know that the X chromosome is a lot longer than the Y chromosome, but the Y chromosome does have regions on it that are said to be homologous with the X chromosome. Specifically, the ends of the Y chromosome are said to be homologous with the X chromosome. So what does this mean if the X and Y chromosome aren't necessarily homologous, but they have regions that are homologous with one another? Well, that simply means that the Y chromosome at the top and bottom of that Y chromosome contains the same genes that are found at the top and bottom of the X chromosome, making them homologous. So why do the X and Y chromosome have to be homologous? There is an important reason why they have to be homologous. So think about what process is used to separate the X and Y chromosome. Hopefully you're thinking of meiosis. When we form gametes, we don't want the X and Y chromosome to go together into the same gamete. We want them to be able to segregate from one another. So how do we get them to segregate from one another? Well, in prophase 1 and metaphase 1, we need to get them to line up together somehow, and it's these homologous regions of the Y and X chromosome that allow them to actually pair up so they can properly be separated during anaphase 1 of meiosis 1. So after fertilization has occurred, and we have that zygote starting to go through mitosis now and make copies of itself, new cells, the zygotes, male or female, depending on the XX chromosome or the XY chromosomes, all the gametes, all the, I should say zygotes, are really all the same up until about 2 months of development. And that's when we'll start to develop anatomical signs of gender. Prior to this, the gonads in males and females developmentally looks identical. You can't really tell them apart. And it's the SRY gene that's found only on the Y chromosome that allows for the distinct change in the development of males. The SR gene is first identified in 1990. It's found on the Y chromosome and is short for sex-determining region of the Y. So sex-determining region would be the SR, and then of the Y would be where that Y comes in the SRY. If there is no SRY gene, the gonads develop into ovaries. So it kind of makes fairly good sense that the SRY gene is found only on that Y chromosome. So let's talk a little bit more about that Y chromosome. First off, the Y chromosome contains 78 genes that we found on it. And of those 78 genes that are functional, 25 proteins are produced. So how do we have 78 genes and only 25 proteins produced from it? Shouldn't be 78 genes and 78 proteins. Well, the reason why is that some of those 78 genes produce the exact same protein. So there's going to be several genes producing the exact same protein. So there is a bit of overlap there. Also, that half of the genes are expressed in the testes for normal testicular function. So most of those genes on the Y chromosome are used strictly for functionality within the reproductive system of the male. The X chromosome is a little bit different from the Y in terms of functionality, and we'll be talking about that in a little bit here. Now, talking about sex-linked genes. Sex-linked genes are genes that are found exclusively on the X chromosome or on the Y chromosome, but not on any of the autosomes. Hence, there are genes that are tied strictly to those sex chromosomes, which affect males and females a little bit differently. There are very, very few disorders linked to the Y chromosome since there's so few genes on it. We said there were 78 genes on it. So there's not many. If a male does have a disorder on the Y chromosomes, his sons will also have that disorder. What about his daughters? Well, they won't have it because they won't get the Y chromosome from him. Otherwise, they would be his sons and they would have it. The most predominant example of this would be that of irregular sperm production, which usually is resulting in low sperm count. So this X chromosome, the human X chromosome contains around 1,100 genes, and that's much more than 78 genes. Thus, there's more disorders linked to the X chromosome because there's more opportunity for disorders to be linked to the X chromosome. And as mentioned previously here, the effect of an X-linked disorder can affect males and it can affect females, but the way that it affects males is very often different from how it affects females, and that's what we're going to be looking at next year. So, X-linked genes and inheritance. As we had mentioned, the Y chromosomes helps determine gender, development of the gonads in the second month of development. X chromosome, has a lot of genes on it that aren't necessarily related to sex. And if you think about it, it kind of makes sense here because males, we have an X chromosome too, but we don't develop any of those secondary female sexual characteristics. So there are some traits on that X chromosome that are expressed in males, but they're not necessarily sex-linked. X-linked genes in humans follow the same pattern of inheritance that Morgan saw in those flies we were looking at previously. And what was found is that fathers passed X-linked alleles to all of the daughters, but none to their sons. And mothers passed their X-linked alleles to both daughters and sons. So we're going to be reflecting on Morgan's experiment here and thinking about that. And What was the soonest that he could see a female with the recessive phenotype? I want you to maybe go back and take a look at the picture tied to his experiment. And you can find that on page 289, figure 15.4 specifically, to respond to the question that's about to pop up on your screen right now, asking you to look at the P generation, the F1 generation, so on and so forth. When is the soonest that a female with white eyes could arise? So look at that experiment, and then respond to the question that's about to pop up. So looking at traits, we'll speak generally about traits here on the X chromosome. Keep in mind that females have 2 copies of the X chromosome, whereas males have 1 copy of the X chromosome. So in terms of recessive traits linked on the X chromosome, females will only express that recessive phenotype if she's homozygous for that allele. And that's nothing really different from what we have been looking at with simple dominant and recessive traits, the Mendelian genetics, but it's a little bit different for males. For an example for females, however, we'll see an X chromosome here and an X chromosome here, with the recessive allele attached to this one and the recessive allele attached to this one. In this particular case, females will express the recessive phenotype. For males, it's a little bit different. Since we only have 1 X chromosome, in order to express the recessive phenotype, we only need 1 copy of that gene. So this particular male, XaY, he's going to express this recessive phenotype fully, whereas females, they'll need 2 copies of that allele. Since males can't really be homozygous in this case, nor heterozygous, they're said to be hemizygous. So we're going to look at a few examples of X-linked disorders, which happen to be recessive. The first one is muscular dystrophy. It affects 1 in 3,500 males, and it's characterized by a continuous weakening of muscles over the course of life. And as a result of muscles being weakened, you tend to lose coordination, start stumbling a little bit. And affected individuals usually don't make it to age 20. They die relatively young. So that's muscular dystrophy. We're going to be talking about another one here in a second. And this one happens to be hemophilia, which is the bleeder's disorder. Essentially what hemophilia is, it's an inability to form blood clots because your body is not producing a certain protein necessary for coagulation or blood clotting. Hemophiliacs, they got to have a rough life because a bruise can be quite serious. You take a slug to the thigh, and you'll develop a bruise yourself. But how does that bruise stop from getting bigger and bigger? Well, it has to clot at some point. And with hemophiliacs, a simple bruise can cause really death because their bodies might not ever stop that blood from leaving the blood vessels within the bruise. This happened to be a common disorder within the royal families of Europe in the 1800s, which was made even worse with the result of cousins marrying cousins. And while that seems kind of disgusting, and I guess it is, they kind of had a limited selection of mates because royalty had to marry royalty. So who's royalty? Other people you're related to already. So that's what went down in the 1800s with the European family. There's pedigrees out there on that one. And there's stories and stuff as well that kind of have made top headlines as of late. I don't think we'll get a chance to talk about those, though. So today, hemophilia, well, still a disorder. It can be treated by hemophiliacs receiving an injection of those clotting proteins that their body can't produce.